This method allows to generate pure, assembly-competent tubulin with defined post-translational modifications in quantities sufficient to perform entire sets of in vitro experiments.
This protocol, based on cycles of tubulin polymerization and depolymerization, is easy to perform in any cell biology lab. It allows to generate good amounts of tubulin with very little hands-on time.
To purify tubulin from suspension cultures, 9 days before the tubulin prep, revive and amplify the desired cell type to obtain 6 confluent 15-centimeter dishes. 8 days before the tubulin prep, add 1 liter of pre-warmed medium to every spinner bottle per dish under a cell culture cabinet and place the spinners on a stirring table inside the cell culture incubator. Leave the lateral spinner caps slightly open to allow the medium to equilibrate to the atmosphere of the incubator. The next day, use the cells from the confluent dishes to inoculate the spinner bottles. Return the spinner bottles to the incubator for 1 week with the lateral valves slightly open. To harvest the cells, transfer the cultures from the spinner bottles into 1-liter centrifuge bottles and collect cells by centrifugation. Resuspend each pellet in 10 milliliters of ice-cold PBS and pool the resuspended cells in a 50 milliliter tube for centrifugation at 4 degrees Celsius. For a 10 milliliter cell pellet, add 10 milliliters of lysis buffer to the pellet